Thank you. Dr. Burgess yields back. The chair recognizes Mr. Cardenas for 5 minutes.
Thank you, Chair Guthrie and Ranking Member Eshoo for holding this important hearing, and I also want to thank the witnesses for sharing your expertise and your opinions with us this morning before the American public. Providing relief and life-saving help to people who have had little reason for hope is ultimately what this is all about. That's why I'm thrilled to see bills that I support up for discussion today, including the Benefit Act, which would require the FDA to consider relevant patient experience data in their new drug approval process, and the Accelerating Kids Access to Care Act, which would ensure that kids can cross state lines and retain coverage for specialist services to treat rare conditions. I'm also glad to see that Give Kids a Chance Act on the list of bills today as well, since it will ultimately improve the potential for treatment options for pediatric cancer. These bills will make life-altering improvements for those who have had every reason to believe their suffering was inevitable. I also want to thank Representative Eshoo for her leadership and innovation on pediatric populations, and it's an incredible legacy to leave to ensure that kids with cancer and rare diseases have hope to treat their conditions. So thank you to our colleague, Anna Eshoo. My first question is for Dr. Bassuk. In your opinion, as a pediatrician, what kind of impact would policy like the Give Kids a Chance Act, which would authorize clinical trials for combinations of drugs to treat pediatric cancer, have on families who feel they have exhausted all of their options?
So I don't know the exact details of the bill, but I will just say the following. Before I was born, if you were a child with leukemia, you had a 90% chance of dying within 5 years. By the time I finished medical school, you had a 90% chance of surviving 5 years later, and that's because of research. Last time I was on call, I was called to see a baby with a brain tumor, and I had to do something that sometimes pediatric neurologists have to do, which is a brain death exam, because we have no good treatments for brain tumor patients. So there are clearly cases where we need to do more than we're doing, and we need to do everything that is available to us, and anything that gives us that kind of ability is something we're all going to support.
Okay, thank you. I also want to discuss the importance of using the most humane methods possible to test new innovations and therapeutics. This means we must move away from animal testing where and if at all possible. To be clear, this transition is not only an animal welfare position, but an imperative to advancing human health as well. We know that approximately 90% of drugs fail in human tests after going through animal trials. In some cases, there is reason to believe that non-animal methods are actually more effective, and failing to phase out harmful animal tests where possible leaves new innovations on the table. I was glad to see the FDA Modernization Act 2.0 get signed into law last Congress. I appreciate efforts to try to implement those policies. Most importantly, though, we must ensure that these efforts are safe and effective for the populations they are intended to treat. Dr. Flott, you mentioned in your testimony that, quote, clarity on regulatory expectations for alternative testing methods is crucial, and research, development, and science-based adoption of such alternatives should be prioritized, end quote. Can you say more about how FDA can speed effective new alternative methods to be more utilized?
Thank you, Congressman Cardenas. The issue is a complex one, of course, because when certain new modalities, basic platforms of gene and cell therapy come to the fore, they require some level of animal testing in order to identify previously unanticipated adverse effects. So there is not a 100% substitution. However, when a drug or biologic is approved, nowadays many of them are approved for a certain set of mutations in a gene. And if that span of mutations can be expanded using the same platform by using cell-based testing methods as opposed to animals, that will, as you point out, speed the additional indications. So it is not an all or none issue, but it is clear that substitution methods can be faster.
Thank you so much. My time expiring, I'm going to be submitting some questions to the panelists to answer in writing in due time. So thank you so much, Mr. Chairman. I yield back.
Gentleman yields back. Now recognize Mr. Latta for his 5 minutes of questioning.
Well, thank you, Mr. Chairman, and thank you very much for our witnesses for being here, and Ms. Davis for your testimony today. We appreciate you being here. One of the privileges of serving on this committee is that we see firsthand, years in advance, the cutting-edge American innovation and what we can do to market those new treatments as soon as possible, especially those when we're talking about rare diseases. This committee has a long-standing history of working in a bipartisan fashion, and I'm proud of the work we've accomplished in this Congress and in the years past. However, I do believe our innovation to cure rare diseases could be at risk as a result of the Inflation Reduction Act. Medical breakthroughs have been made in treating rare diseases, such as cancer and Alzheimer's, and I fear that all the progress accomplished might be in jeopardy and hope lost for those seeking cures. We can and must come together to protect research into rare diseases. And also, again, I want to thank all of those who have been in the research and development of these life-saving drugs and what they bring to market to help so many people across this country and across the globe. Dr. Chen, innovation in drug development has come a long way, and that includes small molecule drugs. When we think about small molecules, what comes to mind are pills that treat prevalent conditions in large populations, easier to pioneer and easy to manufacture. But thanks to robust innovation, the easy next therapies have largely already been developed. These days, small molecule drugs require cutting-edge science with longer development timelines and complex manufacturing requirements. But they hold immense promise for patients, especially those with rare diseases. How do we ensure incentives exist for the development of the groundbreaking small molecule drugs and the new paradigm established by the IRA?
Well, I think we need to make sure that prices match their value. If you're going to put a price control on a high-revenue drug and that high-revenue drug is a high-value drug, then all of a sudden incentives for the high-value drug have been reduced. And so that, I think, will hinder our ability to see the drugs that produce the most value come to our society.
Thank you. Dr. Flott, drug development is an iterative process, especially for oncology drugs. Often the majority of research in cancer therapies happen after the initial approval, including in earlier stages of cancer, and the overall rate of innovation accelerates over time. While the Orphan Cures Act is a good first step, how can we ensure that there are incentives for continued innovation so that orphan indications for such things as life-saving cancer drugs that typically occur throughout the innovation life cycle continue to come to market?
Thank you, Congressman Latta. It's a complex question, but it certainly would reiterate that many of the incentives that we have talked about to speed regulatory review, to broaden the protections that orphan drugs have, and in some cases to define orphan conditions based on specific mutations, as in some types of cancer where globally a type of cancer may not meet the rare definition, but cancer due to one particular mutation may meet that definition. So in a variety of ways, we work collaboratively with the FDA, but I think any legislation that enhances the investment environment so we can get more private industry funding for these types of treatments is essential right now.
Okay, let me follow up. What diseases might be most positively impacted by the passage of the Mini-Act? And what would be negatively impacted if it does not pass? The ASGCT, as discussed, doesn't have a particular position on the Mini-Act. There are a subset of the treatments in our catchment that are based on oligonucleotides, that is, synthesized short stretches of DNA or RNA, such as one of the treatments for SMA1, the Spinraza treatment. Those, we feel, if they're treating rare disease, ultra-orphan diseases, should receive favorable treatment and advancement. Well, thank you very much. Mr. Chairman, my time is close to expiring. I yield back.
Gentleman yields back. Now recognize Ms. Dingle for her 5 minutes of questioning.
Thank you, Mr. Chair. Last summer, I was contacted by the medical director of the Washtenaw County Tuberculosis Clinic, who was treating a patient, Chandra I. Zinga, for a severe post-surgical tuberculosis infection. After a month battling a severe TB infection in the intensive care unit, she died. Sandra's physician was particularly alarmed since he realized she had recently received a bone graft that was manufactured by Uzio Biologic, a company with a troubling history with contaminated bone grafts. In 2021, units of the contaminated bone graft manufactured by Uzio Biologic were implanted into 113 patients. 87 of them developed TB infections and 8 died. Since Chandra's passing, it's been discovered that her death was indeed linked to contaminated bone graft material produced again by Uzio Biologic. She was one of 36 patients who received material from the contaminated lot, and as of today, this latest outbreak is linked to the deaths of 2 people, including Chandra. I'm grateful that the chair included the Effective Screening and Testing for Tuberculosis Act and the Chandra I. Zinga Human Cell and Tissue Product Safety Act as part of today's hearing. I'm leading these 2 bills along with my friend and colleague, Representative John Molinar, who was here, and he is joined by Chandra's sister, Taryn Brunick, who also happens to be John Molinar's staffer. I want to thank you, and we all honor you today and grieve for your sister. So John and I and all of us have been working closely with the Food and Drug Administration and the Centers for Disease Control and Prevention to better understand the manufacture and regulation of human cells and tissues to ensure they can be used to improve patient outcomes, not harm them. Dr. Flott, does the use of human cells, tissues, and tissue-based products support your practice in caring for patients with rare disease?
Thank you, Congressman. There are a variety of different kinds of cellular products, some of which are combined with gene therapy, as Dr. Basouk mentioned, with the treatments for sickle cell disease require a cellular therapy with gene alteration or gene editing in order to be able to be used. What your experiences point out, though, is that there are a wide range of cell therapies out there, some of which combine with gene therapies and some of which do not. And the regulatory science has to continue to advance in order to find out a better test for these unanticipated consequences, such as the one that you talked about.
So we've got to stop them. We don't want to see anyone else die. How can we maintain the integrity and safety profile of these products?
I don't know the specific case in sufficient detail to really address that particular one, but I think the point is that our regulatory frameworks, our testing for quality control cannot be static. When new potential dangers are identified, the quality testing for the treatments has to be updated and improved.
So finally, do you think the American public understands the risks associated with bone grafts and other donor materials? Do you think people who get a bone graft from their dentist or a bone graft for their back think, I could die from this bone graft?
Well, I certainly don't have any data upon which to base an opinion like that. I would just simply state that right now it's certainly within the purview of the provider, of the physician or the dentist to, as accurately as possible, summarize the risks and benefits. But I don't have any data particularly on how well-informed patients are.
I suspect patients aren't told at all. So I want to thank you. A few years ago, people on this committee are tired of hearing about it, but I had multiple bone grafts because of osteomyelitis in my jaw, and no one ever gave me the risks. And that's why these pieces of legislation are so important. Taken together, they will take steps to ensure patients better understand the risks associated with bone graft materials and also improve testing and screening of tuberculosis in donor materials that are used in medical procedures. I look forward to working with the members of this committee and my dear friend Rep. Molinar to ensure we get these bills right. Thank you, Mr. Chairman, and I yield back.
Thank you. Thank the gentlelady. Yields back, and the chair recognizes Mr. Griffith for 5 minutes for questions.
Thank you, Mr. Chairman. Dr. Chen. I come from a little town in Southwest Virginia, but I grew up with a family that was afflicted with Huntington's disease. And since that time, also know another family that through an adoption where they had no way 40 years ago of knowing what was going on, that family now has learned that it also has Huntington's disease. So in your testimony, you mentioned there are targeted therapies that directly address the underlying genetic causes of diseases such as Huntington's and ALS. What are these therapies, and what does the future look for them?
You know, there are a lot of therapies out there that are still being developed, and the targeted therapies in particular are there to address, as you've mentioned, the underlying genetic mechanisms. And as we learn more in innovation and move towards sort of cell and gene therapy, I think we have more potential curative treatments down the road for us.
You know, the situation, particularly with Huntington's, is so dire that one member of that family has told me that he doesn't want to be tested. He's afraid to find out whether he has the disease because there's nothing that can be done for it. That said, do you think that some of the provisions of the Inflation Reduction Act, particularly the confiscatory and what I believe and have argued longly, unconstitutional provisions as related to the fines if you don't cooperate with the federal government and negotiate the way they want you to, do you think that will hinder or enhance advancement of therapies for people facing these types of rare diseases?
You know, the IRA has a carve-out for drugs with a sole indication for a rare disease. The challenge here is drug manufacturers who enter this space consider the entire pipeline of indications that they might get approval for. So what the IRA does is essentially disincentivizes drug manufacturers from entering exploring a drug where the first indication is a rare disease. And it also further disincentivizes drug manufacturers from exploring further follow-on indications, which is a very cost-effective way of doing R&D for the rare disease space.
So let me see if I can translate into layman's terms. If you're a manufacturer of medications and doing the research and you have one that treats a genetic disorder and some of your think tank team says it might help Huntington's, you're afraid to try it because then that would trap you into possibly having confiscatory fines or penalties put on you by Medicare or Medicaid. Is that correct?
That's exactly right. You are potentially subject to price negotiations.
So you're better off if you don't look. Just keep it as the one disease. Okay. Dr. Flop, Huntington's disease, as we've discussed, is an inherited disorder. In your testimony, you mentioned the adeno-associated virus, which is a vector that targets specific cause of a disease and changes the way the cell functions. This is used to treat inherited diseases without treatment, which Huntington's would be. Are you aware of any efforts to use this or to research this related to Huntington's disease?
Yes, Congressman, there are. In fact, there have been several proof of concept studies using AAV to deliver a form of RNA interference. Huntington is a disease where you have to silence a bad gene and not simply add back a good copy of the gene. So there was, in fact, two efforts that I'm aware of. One was actually at University of Iowa by Dr. Beverly Davis using AAV to deliver this technology. The other is a series of studies being done at our institution at UMass Chan Medical School under the leadership of Dr. Neil Aronin, who is one of the people pushing the envelope forward for both gene-based therapies, that is recombinant viruses, and oligonucleotide-based therapies, that is synthesized stretches of DNA or RNA, that either one of which is designed to downregulate that offending gene.
So let me ask you the tough question. If it's your friend who's afraid to get the test to find out whether or not he's carrying that gene for Huntington's, do you tell him that the new treatment looks promising enough to get the test, or should he just go on living his life in somewhat, he's always worried about it, but more blissful because he thinks that he probably couldn't handle the stress
Well, Congressman, unfortunately, I've been asked this question at different times, not specific to Huntington, but to many of the diseases that affect kids that I treat. I have learned not to make any guarantees, but I will say there is reason to still have hope for conditions like this, because if we do what we can do, industry does what they can do, the foundations do what they can do, I am convinced we are going to beat most of these diseases in the end.
I appreciate that. My time is up. I have lots of questions for the record. I yield back.
Gentleman yields back. The chair recognizes Dr. Ruiz for 5 minutes for questions.
Thank you, Mr. Chairman. As an emergency medicine physician, I have treated patients of all ages with various health conditions. I have seen firsthand that there is unmet need for patients struggling with rare diseases. We need to expand access to care for patients with rare diseases. We need to prioritize innovation so we can develop new and potentially life-saving treatments. And above all, we must not leave behind our most vulnerable patients, kids. That is why I co-sponsored the bipartisan bill, the Give Kids a Chance Act, introduced by Rep. McCall and Rep. Eshoo. This bill would provide for clinical studies of combinations of 2 or more cancer therapies for pediatric cancer. Studying combinations of therapies can help lead to new innovative treatments for children with cancer to help them achieve better health outcomes and live longer. Dr. Basu, could you discuss the value of combinatorial studies of targeted cancer drugs for children with cancer?
Yeah, this is actually an easy one. If you look at any individual drug and test it for cancer as a sole therapy in kids, they're all going to fail. We know this because, if you look at adults, they have combination therapy and it's approved. We have to do combination therapy in kids. The cancer cells are smart. They figure out what's going on with the drug and they find a workaround. So that's why you got to gang up on them and use multiple drugs.
Dr. Flatt, in your testimony, you discussed the importance of clinical trials and challenges they face. Do you see a quick path from bench to bedside? Yeah.
The path from bench to bedside needs to be faster than it is now. There are many different ways to do that. Right now, certainly part of that is just the total amount of investment in this from all the parties, as I mentioned before, because these things take money and time really to know the real safety and efficacy. But I would point out that very often, as we talked about before, the window for an individual patient may close while these final stages of testing are being done. So anything we can do to speed approvals or speed entry even into the clinic is important. And one of those concepts is this platform technology, which we as a field are just working with what that means. But basically, if something works in a given cell, to be able to substitute a different gene for patients who have a different but related type of condition.
And do you see companies embracing pediatric studies?
Well, it's an interesting question. Companies do embrace pediatric studies more than they used to, having been in this field for a very long time. However, I will say that there is a lot of risk for investors and for companies in this whole field of cell and gene therapies. And so incentives to offset the risk are truly important and remain very important.
So ensuring patients have access to effective treatments is vital. But we also need to think more broadly about how both innovative treatments and early detection of cancer can save lives. While it is not under consideration by the committee today, I would like to reiterate my support for another bipartisan bill. Mr. Chairman, I would like to reiterate my support for another bipartisan bill that I am an original co-sponsor of, H.R. 2407, the Nancy Gardner-Soul Medicare Multi-Cancer Early Detection Screening Coverage Act. This bill would increase access to multi-cancer early detection tests for our nation's seniors. Dr. Basu, can you talk briefly about why access to early detection and innovative treatment could help to reduce the burden of cancer for the most vulnerable, children and seniors, and how can increased access to clinical trials, early detection and treatments help in reducing health disparities and cancer inequities?
Yeah, I mean, as has been discussed, I mean, many of these things, if we don't detect early, the horse is out of the barn and we can't do anything for the patient. So early detection is absolutely key, and we're very interested in anything that helps with that. To the question of expanding access to therapies, expanding access to clinical trials, some of the changes that this committee has made in the past, and some of the ones that we're talking about today, have already had effect. So Iowa's a small state. We only have 3 million people. And because of that, we have, in the past, not generally been a site for phase 1 clinical trials for cancer. But now we have, just in the last year, because of the laws that we're talking about, we are doing phase 1 clinical trials for kids in Iowa. In Iowa, they don't have to go to the East Coast. They don't have to go to the West Coast. And that is because of this kind of legislation.
Thank you. I urge my colleagues to support both HR 3433, HR 2407, and I urge committee to include these bills in any healthcare package that is being worked on. Thank you, and I yield.